Thank you, Dr. Sherwan. I'm pleased to participate in another omics conference. I'm not talking about stem cells either at this point. I'm talking about cancer cells and cancer cell biology and the interaction of thyroid hormone with cancer cells. Thyroid hormone exists in the circulation primarily in two forms. At the top of the slide is LT4, the principal, L-thyroxine, the principal product of our thyroid glands under normal circumstances. T3 has one iodine missing, and that is the principal form of thyroid hormone inside the cell, acting genomically in the nucleus, but also on mitochondria and on the endoplasmic reticulum. At the bottom of the slide, you see another form of thyroid hormone, which occurs naturally, tetrac. It's derived from T4. It has 4 iodines, but you can see the alanine side chain has become an acetic acid side chain. This is an interesting change. What this has conferred on this molecule inside the cell, it's a thyromimetic. It acts like a T4 or T3, but is lower potential, lower potency than T4 or T3. But on the outside of the cell, where we've defined a receptor, it's an antagonist of thyroid hormone. And where is that receptor on the outside of cells? It's on an integrin. And integrins are, as many of you know, are a couple of dozen of heterodimeric structural proteins that are absolutely crucial to cell-cell and cell extracellular matrix interactions. Those extracellular matrix proteins include vitronectin, fibronectin, and osteopontin, and you'll hear more about osteopontin tomorrow in tumor cells from Dr. Weber, who's up on the panel on the dais today. Recently, integrins, or at least one integrin, alpha V beta 3, has been shown by us and a couple of dozen other labs to bind small molecules. These small molecules include thyroid hormone and its analogs, resveratrol, an interesting compound which has had a checkered history in the pharmaceutical world, and dihydrotestosterone. The thyroid hormone receptor that we've defined on the extracellular domain of alpha V beta 3 binds LT4 very nicely. It binds LT3 less well, and it binds tetraiodothyroacetic acid, tetrac, extremely well. Tetrac blocks, therefore, the binding of T4, also blocks the binding of T3 to the integrin, and it has an array of very interesting anti-angiogenic and anti-cancer properties at alpha V beta 3 that are independent of blocking T4 and T3. So we thought that the actions of tetrac were limited to simply blocking T4's action at the integrin. In fact, in the absence of T4, in the absence of T3, we see a variety of effects of tetrac on cancer cells. Here's a schematic of an integrin for you. It's obviously a heterodimer here, the alpha and beta monomers present. And you can see that there are feet or tails inside the cell that create messages from proteins like osteopontin and vitronectin that are very specific inside the cell. There's a transmembrane portion, and then there's a large 75–80% of it is extracellular domain. And that red X up there is where T4 binds, as defined by X-ray crystallography modeling done by our colleagues at the Hauptmann Woodward Institute in Buffalo. And you can see that there's plasticity to this molecule. This is X-ray crystallography modeling done by Arnaud and his group at the Mass General Hospital 10 or 12 years ago. And you can see that this integrin, any integrin, but this was done with alpha V beta 3, can bend over on itself, or it can straighten itself out in what's called an activated form. And because there are binding sites, recognition sites on this for a variety of proteins and for small molecules, when the shape of the protein changes, it changes the exposure of the binding sites, and thus the activity of this protein and the messages that it can create inside the cell can be highly variable depending upon the state of the integrin physically. How we define the thyroid hormone receptors on the integrin, as shown here. We did binding of radio-labeled hormones to purified integrin. We did siRNA knockdowns of the monomers. We did integrin antibodies probes. We did crystallographic modeling, as I suggested. And then we've also looked at nanoparticulate formulations of thyroid hormone, where we have covalently bound tetrac to a nanoparticle so that it can't get into cells, and have shown that it has activity at the cell surface. So what are the actions of T4 at the receptor in terms of cancer cells and dividing endothelial cells? Well, the hormone acting at the integrin is proliferative, it's anti-apoptotic, and pro-angiogenic by multiple pathways. If we're healthy, it's good to have that anti-apoptotic activity. If we have a cancer on board, we don't want anti-apoptotic activity to be induced by thyroid hormone. The receptor turns out to be a little more complex than we had initially anticipated. It has two domains. It has a domain that binds T3 exclusively, the so-called S1 domain. That has a set of functions. There is the S2 domain, which binds both T4 to a greater extent than T3, binds both T4 and T3. And that has some effects in terms of, different effects in terms of gene transcription. And you can see, however, that it's the S2 domain that is important to cell proliferation. What are the effects? What is a sampling of effects of LT4 on tumor cell gene expression that are initiated on the outside of the cell? This is almost robotic kind of activity where the hormone binds to the plasma, to this protein in the plasma membrane, and in fact that binding results downstream in a variety of effects. The fibroblast growth factor 2 gene is turned on. HIF1 alpha gene is turned on. The COX2 gene is turned on. ER alpha, the estrogen receptor gene. Yeah, this is thyroid hormone turning on the estrogen receptor gene. It turns on the thyroid hormone receptor genes in the nucleus as well. It turns on caspase-3, it turns on NOXA, it turns on iNOS. This is a remarkable set of credentials for thyroid hormone acting on the outside of cell. And we've recently reviewed this, all of these actions of T4 in the published literature. Now, so if that's the case, if T4 has this kind of an effect on tumor cells, what happens if clinically you take T4 away from patients? And some of that has been done by a variety of investigators. Reduction in host circulating T4 levels in patients with advanced cancer may prolong survival in breast cancer, and that's been shown by MD Anderson studies of their experience with spontaneous hypothyroidism and breast cancer. Head and neck tumors studied at the Cleveland Clinic. Kidney renal cell carcinoma studied in Europe and at the University of Utah. And then recently, Alec Hirschberg, a colleague of ours at the Cleveland Clinic, has induced hypothyroidism, euthyroid hypothyroxanemia in patients, and has shown improved outcomes. Now, the way this is done is to give a drug that's a thioamide methimazole that blocks T4 synthesis by the thyroid, so the patient can no longer make T4, and the patient is given T3 to keep him or her happy metabolically. And when you do that, you see survival, as published in The Oncologist. This is an observational paper, not a control study, observational paper. You see survival of threefold, 27 months versus 9 months predicted, based on the kind of tumor that the patients have. Perfectly remarkable kind of response, suggesting that T4 is a problem at the level of cancer cells, and T3 is not. But the thyroid hormone analog has a number of anti-cancer, anti-angiogenic properties that are exploitable. So yes, we can do this. We can create euthyroid hypothyroxanemia, and that may improve survival in patients with advanced carcinoma. Can we do something with Tetrac in experimental models? And the answer is yes. And the way we've done that is to take unmodified Tetrac, so that we could exclude its actions inside the cell. We've taken unmodified Tetrac and covalently bonded it via a linker to 150 to 200 nanometer PLGA, polylactate co-glycolic acid nanoparticles. This limits the access of Tetrac to the inside of the cell. It can't get in. Now, by dumb luck, it turns out to be 10 times more potent than unmodified Tetrac in terms of its anti-cancer activities, and it also has a different pattern of genes that are affected in cancer cells as well. So here's a quick schematic of Nanotetrac. We put 4 to 8 tetracs on each nanoparticle in order to get effective activity. Below 3 and above 8, we see decreased activity here, and that suggests there's maybe steric hindrance problems at the receptor on the cell surface. At the integrin, Nanotetrac regulates via signaling transducing kinases, primarily MAP, ORC1 or 2, and PI3, phosphatidylinositol 3 kinase, PI3K, the expression of a variety of angiogenesis-relevant genes and some cancer-relevant genes. The anti-cancer potency of Nanotetrac, as I suggested, is significantly greater than unmodified Tetrac. Well, so what are the effects of L-thyroxine and of Nanotetrac, or Nano-diamino-tetrac? We've called it by a variety of names, on angiogenesis. Here's a chick chorioallantoic membrane assay. The CAM is a lovely assay in which to rapidly look at angiogenesis. It turns out to be an alpha V beta 3 dependent system. What we do is to cut centimeter by centimeter openings in the shell of the fertilized egg and look at angiogenesis over the course of several days. In the upper left-hand corner, you see the phosphobuffered saline. There's a modest amount of natural angiogenesis occurring. If we look at T4, we induce strenuously angiogenesis in 24 hours. And in the lower left corner of this slide, you see an antibody to alpha V beta 3 blocks the effect of T4. These were early studies done in 2006. If we look and compare T4 and T4 agarose, T4 agarose can't get into cells. T4 is covalently bound, just as it can be to PLG nanoparticles. It can be bound to Cephalose or agarose. And when that happens, it's just as effective as T4, unmodified T4. And if we compare VEGF and FGF to T4, we see that they have similar potencies. DIPA is another form of thyroid hormone, which has been of interest as a pro-angiogenic agent. Here's angiogenesis in the CAM with Tetrac added, and you see that Tetrac blocks the T4 effects and blocks the effects of T4 agarose. These are complicated studies, however, because Tetrac is getting inside the cell as well, where it is slightly pro-angiogenic. So we see some slight differences with Tetrac versus Nanotetrac. The anti-angiogenic properties of Nanotetrac involve a variety of substances. They involve vascular growth factor gene expression. VEGF-A and bovine and basic fibroblast growth factor genes are both shut down. Growth factor receptor gene, the EGFR receptor gene, is shut down. Interaction of growth factors, VEGF, BFGF, PDGF, EGF, with their specific cell surface receptors, also downregulated. In other words, the integrin and Tetrac combination or Nanotetrac combination are, in fact, physically talking to adjacent VEGFR and FGFR, those receptors, and in fact disrupting the interaction of the natural ligand of these receptors with the receptor. Nanotetrac also shuts down release of vascular growth, certain vascular growth factors such as BFGF, and then finally it changes the expression of the cancer cell gene thrombospondin 1. No self-respecting cancer will secrete TSP1 because it's anti-angiogenic, but it can be turned on from the cell surface by Nanotetrac. There's also some endothelial mobility changes or motility changes that are induced by Nanotetrac. If we look at this functionally, here is pancreatic tumor angiogenesis measured grossly by hemoglobin content of the tumor. And you can see in the controls, on the left-hand side of each of these panels, a certain amount of hemoglobin content reflecting angiogenesis in the tumor. Triac and tetrac both turn this, tetrac and nanotetrac turn this down dramatically. We have to use 10 times, we have to use 10 times as much tetrac in order to see this effect as we do nanotetrac. And if that weren't enough, in a series of studies we published just a year and a half ago or so, Nanotetrac also affects some other pro-angiogenic agents, and we were completely surprised by this. If you look at bradykinin, if you look at angiotensin II, if you look at lipopolysaccharide, the pro-angiogenic qualities of these substances, these naturally occurring substances, decreased importantly by Nanotetrac. And so what's the extent? How substantial are these changes? How quantitative can we be in terms of the changes induced by Nanotetrac? Well, here is thrombospondin 1 being turned on by Nanotetrac and Tetrac both, and you can see there's a doubling of the messenger RNA that's produced. If we look at thrombospondin, we see about a 40 to 50% increase, at least in some cases up to 100% increase in thrombospondin 1. And we also see a downregulation by 50% or more of VEGF-A. Here are some histologic studies done in glioblastoma, one of the toughest clinical problems we have to deal with. This is U87MG glioblastoma cells in xenografts. And what we've had done here is to have a pathologist look in a blinded way at 400 slides from xenografts, evaluating the amount of apoptosis, necrosis, cell density, and vasculature present in the tumor. I'm just going to concentrate on the vasculature on the right-hand side here. And you can see there's virtually no vascularization left in these xenografts after 2 weeks of therapy. And this is very organized takedown of the vasculature. There's no hemorrhage in these tumors. It's a systematic devascularization of the tumors. Now, what about cancer cells and xenografts? There was some cancer cell data, the apoptosis and necrosis on the previous slide. Let's look forward at cancer cells and xenografts. What happens here? There's downregulation of apoptosis inhibitors. The X-linked inhibitor of apoptosis, XIAP, MCL1, upregulation of pro-apoptotic caspase-2, caspase-3 as well, BCL2 to L14. There's downregulation of catenin genes. There's upregulation of ChIB1. It's a nuclear inhibitor of catenin activities. There's downregulation of 21 of 23 differentially regulated proto-oncogenes and 8 of 9 cyclins. It's a remarkable effect seen on tumor cells. And then there's upregulation of thrombospondin, as I've said. And what's the extent of these changes induced by Nanotetrac? Here you can see in the case of anti-apoptotic BCL2L14, you see a fourfold increase in the amount of mRNA that's abundance that can be detected. And you see Tetrac doesn't affect it at all. Here is XIAP. There's a 50 to 60% decrease in the amount of X-linked inhibitor of apoptosis. In this case, both Tetrac and Nanotetrac are similarly affected. Here is the epidermal growth factor receptor gene, downregulated by 50% or more by Nanotetrac, untouched by Tetrac. If we look at the tumors themselves as xenografts, rather than looking at cells, as the previous slides showed, here we're looking at volumes of tumor. And you can see that Tetrac, at 10 times the dose of Nanotetrac, is effective in reducing the volume. There is, within 3 to 4 days of initiating therapy, a decrease in volume that continues over the course of the 2 weeks of these studies. If we look at IVUS scans, in vivo imaging system scans of Nanotetrac-treated animals and Tetrac-treated animals, you can see in the case of low-dose Tetrac and Nanotetrac at 0.1 milligram per kilogram, The tumors for pancreatic MPANC 96 cells, xenografts implanted in these animals. You can see the red in the IVUS scan is absolutely viable cells. You can see that the Tetrac at that dose is ineffective, and you can see Nanotetrac has a significantly an important effect on reducing viability of cells in this particular model. Medullary carcinoma of the thyroid, a dreadful tumor for us in endocrinology to deal with. Its effects, the effects of Nanotetrac on these xenografts, also important and fairly impressive. Then if we try to combine angiogenesis and anti-tumor effect in the chick chorioallantoic membrane, we get these kinds of effects. Here is Tetrac on cancer cell implants in the CAM. We can grow cancers in the CAM, and they grow spherically, just as they do in vivo in animals. And what you see with Tetrac after just a couple of days of exposure in high dose Tetrac at 1 micromolar, you see that the vascularity has disappeared. And again, no hemorrhage, but very systematic, taking down of blood vessels. And you see fading of tumor cell mass into the background as well. Okay, Nanotetrac has been effective against xenografts of glioblastoma, breast, prostate, pancreas, kidney, variety of lung tumors, urinary bladder, and also, interestingly, thyroid hormone. That is, T4 stimulates thyroid cancer. What about actions of Nano-diamino-tetrac or Nanotetrac on non-malignant cells or other dividing blood or dividing blood vessel cells? Here are some of the studies we've done: monkey kidney fibroblasts, CV1 or 293 kidney epithelial cells. There's essentially no effect of Nanotetrac or Tetrac on the division of these cells. Okay, that's the end of the story. My conflict of interest is I'm the chief scientific officer of a company that is just starting to develop Nanotetrac. The summary: alpha V beta 3 contains a cell surface receptor for T4 and T3. Tetrac and Nanotetrac inhibit agonist hormone actions at the receptor and are probes for contributions of the receptor. That is, a number of other laboratories are using Nanotetrac now from us to look at cancer cell behavior. In vitro, T4 and T3 act at the integrin via ERK or other kinases to support tumor cell proliferation, a variety of tumors, and to support angiogenesis. Tetrac, Nanotetrac block these effects of T4. Acting via the cell surface receptor on alpha V beta 3, Nanotetrac coherently modulates expression of a variety of genes that are relevant to tumor cell survival. Receptor-initiated actions of thyroid hormone analogs also involve modulation of the crosstalk between integrin and nearby vascular growth factor receptors and crosstalk with nuclear hormone receptor proteins, such as ER-alpha. For example, we've looked at an experimental model of ER-alpha dependent breast cancer, and in that setting, thyroid hormone itself is working just as well as estrogen in terms of turning on the estrogen receptor and phosphorylating it very specifically. Okay, the collaborators here: Shakar Musa, the pharmacologist and who has put together Nanotetrac for us in Albany; Hung Young Lin in Taiwan; and Gloria Lin, Heng Yuan Pang in Albany, and a variety of other laboratories. Murat Yeltsin in Turkey is a marvelous veterinarian who has done 200 to 300 animals at a time for us in looking at the effects of Nanotetrac. That's the story for you. Questions? Go ahead.
Could you help me calibrate this? How does these results with Nanotetrac compare to bevacizumab?
I'm sorry, how did
the results that you have with Nanotetrac compare to bevacizumab in the same models?
How did we develop?
No, how does it compare? Avastin is an alpha V beta 3 inhibitor that's more.
With Avastin?
Yes.
Yeah. We have not looked head-to-head at Avastin. Yes.
Thank you very much for your very interesting talk. And my question is, in fact, the action of tetrac or nanotetrac is, in some aspects, it's similar to RGD oligopeptides. And I wonder, if you attach tetrac on plain surface, not on PLGA nanoparticles, but on the surface, if tetrac promotes the cell adhesion, spreading, formation of focal adhesion plaques like RGD peptides. Did you try it?
We haven't tried. We've looked at RGD, a number of RGD peptides, and Nanotetrac is quite different in terms of the genes that are affected. We generally see a shutdown altogether of gene expression. That's useful. With RGD, it's much more selective here, where we're getting turn on of some genes that are really antithetic to tumor cell survival. But we have not done the experiment you described.
So Nanotetrac is purely a membrane phenomenon.
Yeah.
So that you can't get any neuromimetic effect. Am I reading it right?
Yes, it's more complicated than that, because the nanoparticle is in fact degraded in blood. And so the polylactate coglycolic acid, that's picked off as the drug circulates. And what that leaves, the final product is tetrac, the linker, which is 6 carbons and two nitrogens, and 1 lactate and 1 glucose, 1 glycolic acid. And that, while it can get into the cell, is biologically inactive.
So if you wanted to still activate nuclear thyroid receptors, you're going to have to do it with T3.
That's exactly right. And you could, and we think you could be selective with T3. And that is, you could make a nanoparticle out of it that was only 80 or 60 nanometers, which would get into the cell but not into the nucleus, and might go to the mitochondria, for example, if you wanted to stimulate mitochondria, okay, which you want to do, under some circumstances.
So, Paul, my question is regarding the in vivo models that you showed. What was the mode of delivery for the Nanotetrac?
Sub-Q.
Subcutaneous. So it doesn't affect the normal vascularization or any effect on the bone marrow?
No. So we've done, we now have commercial toxicology scheduled.
Yes.
But we've looked at toxicology throughout the mouse, and we found nothing in any organ, including marrow and blood, effect. So we're lucky today.
Yes.
So Paul, beautiful studies, really. This is fantastic. Seems to be extremely interesting agents with translational potential, no question about that. And I realized all your studies were done in a xeno model where you don't have an immune system going. And you're also aware of the fact that the notion out there, any kind of therapy is immunotherapy. Even you use regular chemotherapy, at the end of the day, you have to stimulate the immune system, that the immune system could eliminate whatever residual cancer stays in the body and hopefully establish a memory where you can control recurrences. So this, this Nanotetrac appears to have very useful features, but I'm wondering whether it will be beneficial to test it in an immunocompetent system, where you also look at the effect on the immune system. I would think probably you will be surprised getting a positive effect, and that may even further help out, whatever direct effect of this Nanotetrac, on tumor cells.
Yeah, we agree. I've reviewed thyroid hormone's effect on the immune system in a couple of papers with colleagues in Italy, and there are a lot of effects there, and they're complex. And I just talked. You and I talked briefly about PD-L1, where T4 turns on PD-L1, which would be would be desirable in terms of immune competence in the normal situation, but it's just exactly what you don't want in the tumor to have the tumor being able to turn on PD-L1. So turning it off in tumor cells with Nanotetrac makes sense. And having it turned on by T4 doesn't make good clinical sense. The issue of how well this is going to work in the entirely immune-competent system, we have spontaneous tumor models cooking, and the studies have not been done yet. But I agree that needs to be looked at. Good point.
Great. Any other questions?